I'm just pulling up my notes here on the side, but I wanted to first, thank you all for having me here, and very excited to get started. The uh, First thing I'd like to do is, say that I don't have any major financial disclosures, unfortunately, but I do have one major disclosure, which is that I'm from New Jersey. So take everything that I have said with a grain of salt. As Dr. Kim already told you, I went to Boston University for my undergrad and medical school in a combined BA MD program. It's also where I met my wife when we got married. Um, then went to Houston, Texas for 7 years, where we had our son also. And now my journey's brought me here with you all for the next 2 years, which I'm very excited to start. These are some of the interesting topics that I thought of to discuss today. See some of the things that I'm sure many of you are also interested in. These are some of my last cases from residency. Cavernomas, aneurysms, bypasses were all very um, uh, very exciting cases. But instead, what I'd like to talk to you today about is actually something like this, something that I think is near and dear to all our hearts in a different way. Um, and More specifically, I'd like to talk to you guys about a neuroprotection in acute ischemic stroke. We'll go over some of the background, basically as to the why we should care about this topic, which I'm sure many of you are already familiar with. Go into some of the stroke pathophysiology and history of neuroprotection strategies. I won't try to belabor all of the points and flesh out every single pathway, but just enough to help us understand the topic better, and then go over some of the results from the experiments that I've personally taken part in during my research year, my R25 funded year as a resident. So to start, stroke is obviously a very morbid disease that affects a great deal of the world's population. In the United States, it's the fourth leading cause of death and disability. Most strokes are ischemic strokes, over 85%, and many of them are due to large vessel occlusions, which we are often involved with, for reperfusion strategies such as thrombolysis and thrombectomy, which have really become the gold standard in the last 10 years or so. Still, even with these reperfusion strategies, improved drugs like tenecteplase, improved technologies in thrombectomy, there's still a huge mortality rate and morbidity rate involved with ischemic stroke. And in fact, over half of our patients are still extremely disabled or dead after stroke. This is an extremely busy slide, which is really just to highlight the complex interplay of the processes involved in stroke. Starting with an occlusion, the endothelium breaks down. There are reactive oxygen species that emerge within the brain and immediately affect the microcellular environment, causing neuronal injury, apoptosis, oxidative damage. Astrocytes also, which are trying to regulate the microenvironment closely, become injured and also begin to die. Microglia are activated to try to mitigate the injury, but sometimes often in a sort of feed-forward loop, wind up injuring the brain more, causing and eliciting things like gliosis and other circulating immune cells to penetrate the area and create more inflammation. To try to boil this down into something a little bit more palatable and schematic, I've just highlighted a few of the processes that are involved in a secondary injury of ischemic brain injury after stroke. And so is an excitotoxicity, an inflammatory response, and an oxidative stress that propagate the injury after there's an initial clot and hypoxic event that lead to further cell death, blood-brain barrier dysfunction, and ultimately cerebral dysfunction. To start, the excitotoxicity is basically a breakdown of autoregulation of glutamate at the level of the synapse. That basically relies on macronutrients and oxygen that aren't there anymore. And what winds up happening is that glutamate floods the synapse in many of the very complex networks of the brain, causes the reuptake of glutamate to be inhibited, and also develop a kind of accessory pathway to cell death, or programmed cell death, apoptosis of neurons. The inflammatory response is also triggered immediately, starting with resident microglia. The secretion of cytokines, chemokines, such as ICAM, IL-2, which are visualized there, or IL-6, recruit other circulating immune cells, such as neutrophils, leads to further inflammation and injury response. And it's also worth understanding that microglia polarize during the acute ischemic response. And generally, when stroke occurs, microglia respond in a time-specific manner, often predominating in this pro-inflammatory or M1 phase early on, which secrete other very inflammatory cytokines such as TNF-alpha, and then later predominate in a more anti-inflammatory or pro-reparative phase, or an M2 microglia. That'll be important in understanding some of the later work that is done. And then, of course, the oxidative stress from reactive oxygen species is a powerful driver of ischemic injury that causes direct DNA injury and also triggers neuronal apoptosis. Primarily, the response to oxidative stress is regulated by NRF2, which is present in nearly every cell in the body. But this pathway is often overwhelmed in stroke, and is something that has been targeted as potential treatment option to no avail, as we'll discuss later. Ultimately, what this means is that the ongoing stressors of stroke create this limited time window for intervention, where there's salvageable brain and thus salvageable function over time that becomes diminished. And so we come to our next topic, which is trying to prevent that secondary injury and extend the window for intervention. So neuroprotection is really any therapeutic strategy to reduce the primary and secondary injury associated with ischemia. The timeline of neuroprotection is really in that first few days that seeks to counteract the phase of injury that we just discussed, and is distinct from where or how the brain changes over time after injury, which is more in the realm of neuroregeneration and rehabilitation. So that all sounds too good to be true, and it is. There have been thousands of agents that have been investigated at this point to at the very least prevent injury, to at least act as an ancillary medication to provide an extended window for intervention and reperfusion, but none have been proven to be successful in reducing morbidity or mortality after stroke. And this comes from many, phase 2 and phase 3 clinical trials. These are just a few that were very high profile at the time, including most recently the NA1 trial, which we'll discuss briefly. And these are all sort of within that targeted scheme that we mentioned earlier. So to start, this NXY059, which is a free radical scavenger, was studied in multiple trials of many thousands of patients that were infused with this drug within the first 6 hours of symptom onset, and absolutely no benefit in terms of their ultimate disability or mortality afterward. Again, the FAST MAG trial, which was published in New England Journal several years ago, was again not effective. This was a huge trial looking at 1,700 patients that were given IV magnesium in the ambulance after stroke. And magnesium, of course, is thought to stabilize cellular membranes and prevent excitotoxicity. But in this case, again, no improvement in the outcome. And then, as I mentioned, NA1 or nerinotide, which is an NMDA receptor blocker that is supposed to prevent excitotoxicity, and also that accessory pathway of neuronal apoptosis, was again ineffective at preventing death or disability after stroke. Although in these secondary analyses, there was potentially a signal in patients who did not receive TPA. To come, and we'll look forward to the results. Well, yeah, what I think are the issues with neuroprotection are well, there are obviously probably multiple issues, but there's difficulty in translating these preclinical models to humans. There's just too much variation in people's genetics and the environment that they live in that we can't account for in the lab, that we typically involve cloned animals or very carefully breeded animals. And also in these trials and many of the early data that then became clinical trials, they're done with inconsistent models, perfusion versus reperfusion models, ones that don't mirror our current clinical practice, so that maybe aren't effective when they get plugged into it. And then ultimately, what I think is the problem is that they're one-dimensional strategies. They're targeting these pathways of excitotoxicity, of the inflammatory response, of the oxidative stress response, and trying to see if that helps. But ultimately, stroke is an extremely heterogeneous disease, and it'd be like kind of the same thought process behind why targeted therapies don't help with glioblastoma, because there are just too many other processes involved in the propagation of this disease than just one of these pathways. And so we need a strategy that affects all of them. And one of the things that I was fortunate to be introduced to was a lab at Baylor that studied closely these basically perinuclear proteins called steroid receptor coactivators. And they developed actually also an agonist of these receptors. That's a small molecule that can basically penetrate into any area of the body. They initially looked at these molecules in cancer models, but then later looked elsewhere. SRCs are proteins in a family of co-regulators. Essentially, they're small proteins that exist around the machinery of DNA transcription and translation, in which transcription and DNA transcription can occur without them. And so they ultimately alter the gene expression of the transcription by modifying the transcription complex, and they coordinate families of genes that are often involved in cellular growth pathways. And the best way that it's been described to me is that activating an SRC helps a cell resist stress or the injury response. And importantly, SRCs are found ubiquitously in all forms throughout the brain. And so this was, a very enticing kind of therapy to explore. This synthetic ligand is a very small molecule that can go into or cross cell membranes easily and be delivered in any form. To drive home the point of how SRCs actually work, they are usually activated in response to injury, either from direct signaling of steroid molecules or through transmembrane proteins that develop a cascade signaling response that ultimately activate SRCs. These then go on to modify gene expression in large families of genes that at the end result modulate things like skeletal muscle differentiation, immune suppression, and migration of the certain immune cells into the environment, and then other growth pathways such as cell membrane creation, proliferation, and angiogenesis. One of the reasons why we chose this drug, in addition to look at in neuroprotection, is that there had been a study in mice looking at this drug, which we call 10-1, in a cardioprotection model where a mouse was given an artificial heart attack, essentially, given the drug, and then the mice were sacrificed and the heart was investigated. And so in this experiment, 10-1 treated animals showed a decreased inflammatory response in their myocardium, involved decreased fibroblast remodeling of the heart, and in fact showed improved cardiac function with small echocardiograms that were done. And so this seemed like a promising sort of avenue to explore. And so we went to look at this potential model for SRC mediated neuroprotection. And the experiment was done in rats. This is one that I did in conjunction with actually Dr. Can's lab at UTMB. And we used 20 rats in 2 separate time periods. One day after an acute ischemic stroke was performed, and then at 4 days. The model we used was a middle cerebral artery occlusion model where the rats were anesthetized. Their carotid arteries were exposed in the neck, and we placed a small silk microfilament into the internal carotid artery to occlude the MCA under a microdoppler kind of analysis, and to ensure that the MCA was occluded. And then after 90 minutes, we withdrew the filament and then kind of tied off the opening, and then we gave the drug. And the drug was given again at 24 hours, at 2 days, 3 days, and 4 days. And the mice were sacrificed at those various time points, and behavioral testing was also done. So for the analysis, we'll discuss what I think is the fundamental question, which is, did it work? did the infarct volumes. And to do so, we did what's called TTC staining, which is a common stain used that is stains respiring cells red, essentially. And then we also used the Bederson score, which is also a well accepted and validated scoring system for neurologic outcome in rodents. And then we sought to also determine the changes in phenotype of the other cell types within the brain at the time of injury. And so we used cell-specific markers and things like tunnel assay, which is a kit used to detect apoptosis in neurons and other cells. Just briefly to review the model, which probably many of you are familiar with, we used a microdoppler to supplement a middle cerebral artery occlusion model. Essentially, we started with placing a microdoppler array on the skull of a rodent, which could sense infrared light through the skull, the thin skull of the rodent. And then we flipped them over. We opened the neck here, as you know, I tried to record from one of the microscopes that we use. We go past the very enlarged thymus of the rat. We expose the carotid artery. So you can see here that there's the bifurcation, external carotid artery medially, ICA laterally. And we essentially cut the ECA and we place a microfilament through the cut end of the ECA and then feed it into the ICA. And then we wait for a drop in signal from the microdoppler and we keep it there for 90 minutes. It's kind of hard to see because to make this recording, I had to use only one eyepiece of the microscope. But here's us just setting up the insertion of the silk thread. This is with the CCA clamped. And here's basically us inserting the thread into the ECA going into the ICA. And you can see that thread within the ICA now. And so ultimately, what were the results? Well, it did work. And not surprisingly, but what was surprising was that it reduced the infarct sizes by over 50% at 1 day and at 4 days. So this is a robust finding. You can see some of the representative cuts of the brain here in some of the subjects. The red here is the normal brain structure, and then white being the infarct area. And just looking at these representative subjects in the control and treatment groups, you can see immediately that there's a large difference. And of course, these are just some of the most well-preserved brains that we had, but the stroke sizes were much smaller, and they were also involved less of the cortical areas of the brain. The drug also seemed to preserve neurologic function, and here's the quantification of the Bederson scale. The lower the Bederson score, the better in this case. By 1 and 4 days, the treatment group also did a lot better. Here's an example of one of the categories in the Bederson scale, which is circling. Oftentimes, hemiplegic rats will circle around, and that is a sign of extreme morbidity of the rat after stroke. And as I had alluded to, the cortical infarct areas in the treated brain seem to be smaller. And as you know, as stroke propagates over time, it starts in the deeper cortical areas and expands to the more cortical areas where there are greater collaterals. And so that sort of makes sense with thinking about this as a way of basically decreasing the time of injury of the brain after stroke. Here's a quantification and the between the differences in infarct areas. A very busy slide. Basically, suffice it to say here is that this dark gray bar, which is the treatment group, um, is much smaller than the cortical areas and of the control group. Now, going on to look at the phenotypes of the cells that change with the drug treatment after stroke, we used kind of several cellular markers to identify these cell types using an immunofluorescence model. Essentially, we stained these different slices of brains and then used these average of 5 to 10 high power fields under a fluorescent microscope to do these quantifications. Here's what the raw pictures kind of look like. So we use NeuN, which stains the membranes of neurons, or actually the cell bodies of neurons. And we also use this tunnel assay, which stains for different Cas spaces involved in cellular apoptosis. And we did this at 1 and 4 days and ultimately found that the proportion of tunnel positive neurons, or neurons that are actively undergoing apoptosis, was much lower in the treatment group. Again, this dark gray bar at both time points. And we also looked at microglia. And here we have these beautiful pictures of microglia, which are stained with IBA1. And there's also a DAP stain that looks at just cell membranes here. So these are just all other different kinds of cells. But you can see also the difference between just the pure shape of these microglia, which lend us back to the conversation that we had earlier about changes in microglia cell type as injury progresses. And so to review that, when microglia are activated, they start in this kind of ramified state where they're secreting many inflammatory cytokines, causing increased inflammatory response, to then develop into this amoeboid shape, which then are sort of more participatory in things that are considered pro-reparative, such as secreting growth hormones to induce cellular regeneration, things like that. And so what we found was ultimately decreased, I guess, pattern of these M1 or pro-inflammatory microglia, and an increase and a decrease also in the infiltration of microglia in the late phases of injury. So here again are these raw images, immunofluorescent images, and you can see in this column involving just the control pattern that the microglia are smaller than the treatment group, and then later on also they are less in that M2 phase of their response to injury. And the quantifications show that as well by looking at the intensity of staining of IBA1 and ARG1, which are both markers for these M2 microglia. Another thing we looked at are glial cells, in particular their response to oxidative stress. And so NRF2, as we mentioned, is a pathway involved in managing oxidative stress and reactive oxygen species. You can see that we compared the normal side of controlled and the stroke side of control and treatment animals, and found that the NRF2 pathway was very upregulated within the treatment group after 10-1. And this bore out even by 4 days. And the quantification shows that as well. So ultimately what we found here was that the astrocytes had increased their expression of NRF2 after stroke, that ultimately what this meant was an attenuation of scar tissue or astrogliosis within the penumbra of these brains as well. This is sort of a pathway that's related to, but separate from the NRF2 pathway within glial cells. This is GLT1, which is actually a pathway involved in signaling glutamate signaling. We've also found that, again, this pathway was upregulated within glial cells or within astrocytes, specifically with 10-1 treated rats. Lastly, something that we found and didn't expect to find was an upregulation of regulatory T cells in the core area of infarction within the brain. And T cells, of course, come in different varieties. These are CD4 positive cells that have sort of differentiated into what are considered sort of master regulators of the immune response. And here they're stained with what's a intracellular or intranuclear protein called FOXP3, which is very specific for Tregs. And what this means to me or suggests is that the immune response is also being attenuated. And this is most robust at 4 days and is significantly increased in the treatment group. So in summary, these 10-1 treated animals showed an overall reduction in the infarction volume, which we explored with TTC staining at 1 and 4 days, and preserved neurologic function. So that ultimately was what we tried to prove, and that bore out in a reduction in neuronal apoptotic activity. Of course, why that is could be a number of things, but we sought to at least explore that with how these phenotypes of the various cell types in the brain have changed. And so for one, we found that there's an activation of more pro-reparative microglia after 10-1 treatment, and that there's an upregulation of these pathways involved in oxidative damage and excitotoxicity, NRF2 and GLT1 respectively, within the penumbra, and that there's a recruitment potentially of T regulatory cells within the core infarct that might also reduce the inflammation and lead to a more pro-reparative state again. What I hope that this will ultimately lead to, and going back to the big picture, is that SRC agonists such as 10-1 and really any other neuroprotectant is able to really extend the window for intervention, for reperfusion. And what that might look like is just, essentially extending out the time at which we can effectively give thrombolysis or remove a clot while preserving those critical areas of the brain. One of the things that I sought to do in my R25 year, but what didn't successfully get a chance to do, was to study the changes in gene expression, which I feel are critical in really in measuring the actual response of these treated rats. And this is an example of one of the experiments they did in the cardiac model that actually found this kind of clusters of cells that had a change in their overall expression after treatment. And that's something that I would hope to do in the future as well. And then also looking and exploring the impact of immunomodulation in the acute phase of stroke. overall, I think is not a new concept, but I think one that is key to trying to mitigate the secondary injury of the brain after stroke. In particular, I think that T regulatory cells are important here. There's been a lot of study recently of how circulating T regulatory cells actually get into the brain, and then once they do, what kind of signaling cascades they are involved with. And overall, their presence within the brain within 24 hours seems to be overwhelmingly positive. They lead to things like actually regulating the microglia response, to secreting IL-10 that ultimately creates or propagates the differentiation of neural stem cells. So I do think that that's worth looking into in the future, and just looking at possible ways to do that. There have been people who have injected peripherally polyclonal Tregs either intraventricularly or through the blood, and have found some improvement in inflammatory cytokines after stroke in people even. And another way to do that, of course, is to create a chimeric antigen model, which other people have done for cancer, that creates an even greater immune response, and even more specific, because you can load the membranes of these T regulatory cells with whatever antigen you want. That, of course, begs the question of, what you target, the timing of treatment, manufacturing, etc. But I think that this will be this could play a key role in developing an effective neuroprotectant. Again, I think it's so important to consider neuroprotection in the proper scheme of timing, and in the proper scheme of the continuum of care for stroke. And real comprehensive stroke management, to me at least, is one that involves an intervention at all phases of the injury cascade. And so ideally, what we're looking at is a holistic approach to stroke that includes the rapid administration of neuroprotection agents, and of course reperfusion therapies, and life-saving interventions like decompression, but also later revascularizations and immunomodulatory strategies, potentially, that will help lead patients to the final phase of stroke care, which is neurorehabilitation and potentially neuroregeneration. And I put here some, neuromodulation, brain-machine interfaces as also potential treatment options in the chronic phase. So what I find myself interested mostly in for a research career is to establish a comprehensive model for stroke that integrates acute stroke care, prevention of secondary injury, and neurorehabilitation, that sets a new standard for neurological, neurosurgical care and recovery for better patient outcomes. Obviously, I have a lot of people to acknowledge here. These are just some of the few key people that helped me with my experiments at Baylor, at UTMB, Dr. Can, who some of you might know, in his lab, as well as at Baylor with Dr. O'Malley, who actually discovered these steroid receptor co-activators, and is a very accomplished researcher. Thank you all for your time. I'll take any questions.
Thank you, Tyler, for your presentation. Really appreciate it. I'll open up the floor for questions.
Louis, if I may, I don't know how to use my phone and raise my hand. Sorry. Tyler, beautiful work. A quick question is, what is the natural history of stroke in murine models? Do they, like long-term, do they also similarly have the same patterns of histopathological, like encephalomalacia? What is their clinical? Do they recover from it? I mean, 5 years of PhD, I totally understand that you have to have a working model, but I'm just curious to know how similar is their physiology to a human system?
Yeah, I think that's probably the crux of the issue with these models. Obviously, there have been, it's a very widely accepted model. I think on the surface they're very similar. The circuit, the anatomy of the brain between rodents and humans is similar. They have the same major considerations for your circulation, anterior versus posterior, even neurodevelopment. I think the way that they obviously respond to stroke and the way that you can measure it is much different because, they don't have certain eloquence in their cortex, that really all you can measure are things like their behavior. Do they eat? How do they move? Things like circling, for example, these are all, pathognomonic for severe neurologic injury. But other than that, there's a disconnect between rats and humans in that regard. And the other thing that I sort of alluded to is that the collateral circulation may be a little different. Humans often develop strokes over a long period of time where they have a buildup of atherosclerotic disease or vessel inflammation and have developed collateral such that when an acute event occurs, they may not be as injured, or they may, the stroke may involve a different area of the brain. Whereas these rats are all very young. They're, basically newborn rats in some studies, and they're all, genetically very similar as opposed to humans. So I do think that anatomically and theoretically, rat brains are very similar to human brains, but practically speaking, I think doing these experiments on rats is one of the reasons why all these studies have failed.
Hey,
Thank you.
Tyler. Great talking. Congrats on all this work. One of my questions is, what are some of the systemic side effects of SRC? Because I think a lot of the drugs that try and inhibit the inflammatory response fail because our stroke patients are so prone to infection. And when we modulate the inflammatory system, they all end up doing worse because of the subsequent infection etiology. So what's some of the systemic effects?
Yeah, one thing I didn't highlight well enough probably is that these SRC, these coactivators, are kind of like background proteins, so to speak. So they don't emerge, or they don't get at that. They don't get activated. They don't start to modulate gene activity or gene expression without a stress. And so ultimately, if you just inject this molecule in cells that aren't undergoing a stress response, they won't do anything. And this has been proven by Dr. O'Malley's lab in different models because, as I had mentioned, they were looking at this as a treatment for cancer, for ovarian cancer, actually specifically. And they found that in using this drug in rats and in other animals that didn't have cancer, it didn't do anything. It was practically inert. So I think that it's a drug that could potentially be thought of as only effective for stressed or for a response to cellular stress. And so do we know what will happen in humans? No. And number two, do we know what happens over a very long period of time? No. But I do know that it's a very small molecule. It doesn't stick around for very long, based on the experiments that we've done in rodents, and that it doesn't seem to affect normal cells that are not either not within a growth cycle or have undergoing stress of some sort.
Awesome, thanks.
Any other questions from the audience? I would just, that's the
Dr.
one.
Levitt, I guess.
Oh, go ahead, Dr. Levitt, go ahead.
I'm sorry.
Well, he actually raised his hand. Wow.
I did. I'm trying to be polite. My question is more of a meta question, which is, what makes you think that, or let's step back. Neuroprotection is the graveyard of a lot of talented people, and it's a big problem, and biting off a big problem is often daunting. What's like the next immediate step? Like, what are you going to write your K award application on? It can't just be neuroprotection because that's too broad and not focused. So knowing what you know now, what's the next set of experiments that need to be done to get to where we need to go?
I think there's I guess an ideal situation where I have, free access to collaborate with this group that has a patent on this drug, where I could, move forward with experiments using the drug. And if that's the case, then what I would want to do is look at changes in gene expression with single cell sequencing, as I had planned to initially. I think one of the key things that I've shown here, but didn't necessarily, that I think should be a caveat at least, is that I've only shown you that there's a change in phenotype of different cell types. I haven't shown you that they're actually, their behavior has changed, the way that they've reacted to stress has changed. We know that just from these set of experiments that there's less cortical infarction, which is great, but why does it work? I don't know. And one of the reasons, one of the theories that I have, and what I would write my K award for, is to explore immunomodulation at different time points in acute stroke. And so what I mean by that is, I think, as Dr. Chen had alluded to, if you just shut off the immune system somehow immediately after stroke, you'll have a problem. The neuroregeneration, all the good things that you can think of in the cellular microenvironment also require an inflammatory response. So if you just give them right away something that completely crushes the immune response, they'll do more poorly. And there are some studies that show that. And then long term, if you give them immunosuppressants, you could give them an infection. They can get certain immunodeficiency related opportunistic diseases. So it's all about timing. So I would really want to explore the right time to use a drug like this, for example, to suppress the immune system, and then also to modulate the immune system with things like T regulatory cells. And so what I'd want to do is take the experiments further that have been done on T regulatory cells, both circulating polyclonal T regulatory cells and potentially CAR T regulatory cells to modulate the immune response. Because ultimately what they do is they don't just shut off the immune response, so they don't just activate in certain areas. They're creating an environment that suppresses excessive phagocytosis by macrophages and microglia, but they also inject these growth pathways into the brain by activating neuroprogenitor cells and things like this. So that's kind of the angle at which I'd want to move forward. And I think that it can be torn off into smaller pieces. That isn't just like a huge, like I'm going to cure stroke with T cells, but rather looking at it like, okay, can we at least prevent neuronal apoptosis using these, or can we at least solicit these neuroprogenitor cells to go into the area and start to create new neurons. So
Thank you.
Tyler, I would ask an even more meta question, which is neuroprotection using mirroring models of stroke have resulted in numerous and thousands, hundreds of cures of stroke in mice. We've conquered stroke and in saving the disease in mice, but every translational and clinical trial in primates and humans have yielded negative results. So why continue to pursue this model for elucidating mechanisms of stroke protection?
Yeah, I mean, I think that it's the, as you and Dr. Levitt have pointed out, it's the reason why most people don't pursue the field anymore. But I mean, people still do GBM research, and the needle for that hasn't been moved in over 20 years. So I think the 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 pursuit of neuroprotection is kind of like the holy grail. It's like theoretical, and it's like would be a huge boon to stroke care. Um, Ultimately, there's a great need for it, and you can't just start with people in a lot of these situations. You need to start with animal models. You need to start with fleshing out certain pathways with measuring gene expression, sometimes in vitro models. So I think this is just where it starts. I think it's an important mission, and in this particular case with this steroid receptor coactivator, I think that it's very, it'd be great because it's very safe. So it's kind of like why the Fast Mag trial was so quick to recruit and take off, is because IV magnesium is very safe to give, as opposed to neurinotide or some other things that have had side effects. So I do have hope that, number one, these experiments can advance to humans more quickly. And secondarily, I do think that, just in the realm of neuroprotection, animal models are still very important, even though they're very flawed, as we mentioned.